And with that, I guess we will move on to our next speaker. But thank you again, Dr. Bigler, for a very fantastic overview.
Well, you're very welcome, and I'm going to go see my grandson. So
great to hear that.
So I'll be doing pediatrics here. Different.
Great.
So thank you very much, everyone.
Thank you. So now it gives me great pleasure to introduce one of my colleagues, Dr. David Mikulis, who's a neuroradiologist and professor and co-director of the medical imaging research in the Department of Medical Imaging at University of Toronto. He's a professor and director of the Functional Neuroimaging Lab in the Joint Department of Medical Imaging at the University Health Network and University of Toronto. Primary emphasis of his work is really translational research, focusing on the application of novel imaging methods into the clinical environment. He established one of the first fMRI labs in Canada in 1993, and is currently involved in developing advanced neurovascular imaging methods and major program arms, including quantitative measurement and clinical application of cerebrovascular reactivity, leading to the development of the brain stress test. He also is looking at high resolution and functional imaging of intra and extracranial blood vessel walls that improves diagnostic accuracy for multiple different diseases, as well as discovery of new methods for measuring brain blood flow. So I will leave you with Dr. Mikulis.
Thanks, Carmela, for that kind introduction. So I've got good news. Hello? Can you hear me?
Yes, we hear you.
Oh, so I have good news and I got bad news. The good news is that I have, my presentation follows the world's expert on this area and I feel validated. The bad news is this is going to be a review for everybody from the last presentation. So thanks for that, Aaron. It was really terrific. I very much appreciated it, and it validated, I think, our thoughts along the lines of concussion and imaging it. So I have a disclosure. I use a non-commercial investigational device built by Thornhill to enable, this is going by itself, to enable measurement of cerebrovascular reactivity studies in this, for scientific research. So I'm going to dive right in. Imaging concussion is a subtle injury to a very complex structure with multiple interconnected components. So if we look at an image of neurons in the brain, we see axons, many axons. And this is what the main focus of imaging has been, to understand the structural imagery, particularly affecting axons in the brain. But there are other complex structures. So you can see the red astrocytes in the brain, and they have long processes, but no one, to my knowledge, has thought about how much concussion could injure the glial cells. Then there's additional brain complexity because there are other glial cells called microglia, which react to the injury. And then there are oligodendrogliocytes with long processes that can connect to axons. And these could also be vulnerable to shear injury, but no one seems to be mentioning the effect of trauma on these cells. So there are multiple systems involved that could be affected by the injury, not fully studied. So my particular area of interest is in the cerebral vasculature. And we all know that after traumatic brain injury, blood flow dysregulation is a common feature. And I think this is an area where we can gain some insights. And it also offers an ability in longitudinal studies to determine if dysregulated blood flow persists, does that cause secondary brain injury? And does it have a relationship with post-concussion syndrome? Okay, so I love this Nobel Prize winning picture from Ramon Cajal, who won a Nobel Prize in 1906. And he shows these astrocytes literally embracing neurons and embracing axons. So there's a connection going on here. This is also completely neglected in concussion research as far as I can tell. So let's start off with conventional imaging. We know that as Aaron showed, from CT scanners from 30 years ago, we're not doing much better. We have much better CT instruments, but we still find that 30% of normal CTs in patients with a GCS of 15 have positive MRIs. So concussion falls within the category of what we feel are neurological conditions that have normal clinical MRI scans. And that would include autism, and it would include schizophrenia. So I think this still is debated as to whether or not we should use that as a divider between concussion and more advanced traumatic brain injury, but that's sort of our feeling at the Canadian Concussion Center. And to further that argument, we studied patients with PCS who were diagnosed with concussions by physicians, and of those, 97% of patients had scans that were indistinguishable from the healthy population. So this kind of substantiates the concept that concussion doesn't cause abnormalities in the brain. Now, we did see one thing. We did see these white matter hyperintensities in patients with concussion, but we also saw those in our controls. The difference being that these are slightly more prevalent than in concussed patients compared to controls, but it wasn't statistically significant. And these could represent axonal injury. So if you see them in the absence of brain atrophy or hemorrhage, you can't be sure whether they're related to axonal injury or they're part of normal aging or exposure to vascular risk factors, for example. So what is the nature of the injury that challenges detection by modern imaging? Well, we're imaging these structures. These are microtubules and axons. And we're also, because of that injury, affecting axoplasmic transport, and then you get accumulation of proteins with regional swelling, varicosities in bulbs and axons. I'll show that. We also get injury to other cytoskeletal structural elements, including these neurofilaments, which organize other organelles and proteins in the axon, and they can be released into the extracerebral fluids and detected that way. So axonal injury causes bending and rupture of microtubules, ruptures microtubules. So it interrupts this axoplasmic transport, forming these varicosities that we see on histology, and it gets worse. So if the axon completely ruptures, we see these retraction bulbs. Almost looks like an axon, a dysplastic axon with a synapse sort of coming to the end of it. So why is it so difficult to image this injury? So Aaron clearly alluded to this. This damage is subtle, it's microscopic, and it's sparse. So if you look histologically at what we see, here's an imaging voxel, typically 1 to 3 millimeters. This is a little bit smaller than that, but within that field of view, you see about 7 or 8 of these injured axons. So trying to pick this tiny amount of tissue abnormality in the sea of what otherwise looks like normal tissue is like trying to find a needle in a haystack for us. So the conventional images don't pick that up. So I'm going to talk about imaging versus blood. I hope I have time at the end of my presentation to talk about blood for detecting concussive injury, because there's a link here that I never realized before, literally until last night when I was getting this talk together. So this is hot off the presses. It comes at the end. If Carmela cuts me off, you guys can't hear it. So the current status of advanced neuroimaging, we have the quantitative tools available to detect structural and functional injury. How good are they? So we have advanced imaging, and we can see that advanced imaging has kind of failed us. We have radioactive tracers, we have advanced MRI methods, we have magnetoencephalography, and I'm not going to talk about the latter. That's not my area of expertise, but that also falls into the category of functional neuroimaging. Here's a SPECT scanner. We can put patients into this device. It can look at regional differences in cerebral brain blood flow, but what's the problem? The problem is this: there's regional heterogeneity in healthy controls. So if I kind of blow some of these images up, is this high uptake? Is that high uptake in this control? Is this reduced uptake in the temporal occipital region? The problem is, oops, let me get back to that. The problem is that this is a very subjective assessment, and we need better tools to show what is going on in the brain in patients with injury compared to the healthy variation. So if we look at SPECT imaging and traumatic brain injury, the positive predictive value with SPECT as it's currently performed is only about 59%. It's not good enough. So the SPECT issues are, it's a subjective assessment at the moment from a clinical perspective. And what we need is standardization of the data acquisition and the quantitative analysis. This enables an objective assessment. So how do we do that? Well, oh, and by the way, before I get to that, there's a ruling from the Ontario courts about this. So in 2021, the Ontario Superior Court of Justice was examining this case, and Justice Beal found that SPECT scans failed to meet the reliability foundation test for novel scientific evidence. So we have a problem there. How do we solve the problem? And it's a problem I think we have solved, and it's not that difficult. And what you can do is you can take a large group of healthy volunteers, merge their brains into a common brain space or template, and then you can get a mean and standard deviation for whatever metric you're trying to measure for every point in the brain. And then you can take the patient's scan and determine how many standard deviations outside the normal brain that particular metric is. So here's an example of what they did in nuclear medicine. They did just that. And this comes off of a GE unit. They were looking at blood flow. These red areas were areas in the brain that showed a reduction in blood flow. And this is the table that's generated. So these are the Z scores, and a Z score of 0 obviously is normal. It's the normal mean and with its standard deviation. And if you consider 2 standard deviations outside the range of normal, then you can see in this particular individual, all of these areas in the brain, like 30 or 40 areas, the majority of them are showing a reduction in blood flow. So when you see that, there's no interpretation needed here. You're just looking at it and you say, yes, this patient has extensive reduction in cerebral blood flow, and it's quantitative. The other advantage to SPECT, though, and I don't think we can completely ignore this feature, is that these radioactive tracers can be targeted to very specific biomolecular processes. And they have a very high sensitivity as a result of that, because this radiation that's given off is easy to detect. So this is a study that looked at neuroinflammation. These are the microglia activated after brain injury. And you can see that in controls is kind of this background uptake of the tracer to the microglia. After injury, it increases a bit, and this is PCS, and it's in very high concentration. So then the question is going to become, do we need to build a database to see whether or not patients with PCS have this significant elevated concentration? And this was a very small series. So this is work that needs to be done to make that determination. So other advanced MRI methods, I'm going to talk about what's close to my heart and my brain, or my dilating blood vessels all the time while I'm giving this presentation, and I hope they're doing it effectively. We'll look at cortical thickness. There's a relationship here. If you don't get this, you get thinning of the cortex. So does concussive injury injure vessels and have effects on neurovascular coupling? Diffusion tensor, we've seen nice, elegant demonstrations of how this is useful in looking at axonal integrity. And there's resting state connectivity, which looks basically at how well the brain is connected to itself. So volumetric imaging, we have parcellations of the brain. We're looking in concussion. Are any of these parcellations showing evidence of tissue loss? And we did that. We looked at that. Carmela was the senior author on this study. And basically what we found was nothing. All we did find was that there may be a slight volume change in the hippocampal formation, kind of in the beautiful cone of vulnerability or cone of injury that Aaron has shown, one of those structures. So it fits with that. But again, it's not significant enough to be able to make that diagnosis in an individual. Here's one from blast injury and post-concussive persistence of symptoms. Here's the rate of loss of cortical thickness with normal aging. Here's the rate of loss following concussion. It's accelerated by a factor of 4, but because of this data scatter, you cannot make a diagnosis in an individual subject. So diffusion tensor imaging measures the structural changes in tissue based on how water is moving in that tissue and how the injury alters that water. So if we image celery and we look at conventional imaging, we don't see much. we have the luxury in MRI to determine which direction we want to set our sensitivity to. And when we set it in the right direction and we add diffusion to gradients, we see beautiful tracts in the celery. Of course, I wanted to eat the celery and then put it back in the scanner to see what kind of traumatic injury would do to this, but I kind of ate it and I swallowed it, so it didn't happen. Fiber tracking. Here's a great injury on great demonstration of what subconcussive injury can be seen with diffusion tensor. And here's Aaron's model of torsion between the brain and brainstem. And look what happens to these corticospinal tracts between the brain, midbrain, and going on down into the spinal cord. It's this zone of twisting vulnerability that shows the abnormality in fractional anisotropy, showing disorganization of neurons as a result of that twisting torsion. But it's a group level diagnosis, doesn't work in individuals. The injury is subtle. So that's the theme that's coming out of advanced imaging. Group level diagnosis is possible. Single subject diagnosis, we're not quite there yet. Same for resting state. If you look at these three voxels in the brain, you'll say these two are most likely going to connect. Oops. Rather than the other two pixels. So therefore, you can put the pixels that are well connected and you get the default mode network. What happens after injury? Well, it shrinks in this study. Again, these are group level differences. We can look at more sophisticated analysis of this, which is graph theory. So this is the functional connectome that Aaron was mentioning. And what it does is it looks at hubs and the nodes around the hubs and how many connections there are regionally, locally between these structures. And then it looks at the distant connections. What do we find? Well, if you look at traumatic brain injury, we find that the brain is more connected to itself locally, and the path between these distant connections is longer. Again, group to group findings, not successful in single individuals. So we did a study, Carmela did a study. This is great because we looked at this idea of merging structural and functional analytics. So we had evidence in sports contact sports athletes that had played contact sports, and we found white matter integrity and volume of the corpus callosum that were correlated, and then we correlated them with serum NFL levels. So these are neurofilaments that are released during neuronal injury. That's great. We also saw that there was increasing resting state functional connectivity between the superior frontal gyrus and right hippocampus, kind of in that cone of injury. And it's interesting because some of these studies show increased functional connectivity and some show decreased functional connectivity. I think the research literature is trying to sort out what these differences mean. But the bottom line is, this is still not diagnostic in single individuals. Now, let's go further. Let's unify structure and function. So we have the functional connectome and we have the structural connectome. Let's connect the connectomes. And what do we see? Well, this works, but currently it's only working in small organisms. It's not working in the human brain, which has 100 billion neurons and an average of 1,000 synapses per neuron. So we've got a long way to go to make that happen. I'm interested in looking at blood flow metrics. We do cerebrovascular reactivity. We stimulate blood flow with carbon dioxide in a very precise precision medicine approach. So we can make blood flow change in a very specific way because we have this control. But the problem is we don't have good ways to measure brain blood flow. We use a surrogate of that, but nevertheless, it works. Here's our our control atlas, and then we can take an individual subject. You can see here, this patient's entire brain is 1 to 2 standard deviations below normal. And when we did that in patients within 1 week of concussion, our ROC values at that time were the best of any other test I had seen in the literature. So we have ROC values in single subjects at 0.9 for women and 0.93 for men. And we're looking at both the speed and the magnitude of response. I believe these speed and magnitude responses are biomarkers of vascular performance in the healthy human brain. And we can determine the lack of health in those vessels in various diseases, one of them which is concussion. Now, we know there's autonomic innervation from the brain. It comes from the brainstem. Why on this planet does no one talk about injury to these projecting axons which not only synapse on cortical neurons, but they synapse directly on vessels themselves. So what effect could that have on cortical injury? And it may explain why we're seeing these deficits in patients with cerebrovascular reactivity. And what's even more important is the deficit we see in these patients with CVR studies is that they have a faster response to the stimulus and a greater response to the stimulus. So we have never seen that in any other condition. Every other condition we've studied, cerebrovascular disease, all of these conditions decrease magnitude and speed of response. So this kind of takes out those more comorbidities that affect the brain out the window, and we can actually begin to now say that we have a pretty strong way of beginning to understand this, even in the setting of comorbidities, potentially. So in summary, the advanced neuroimaging status, this is the holy grail, seeing it in single subjects. So the group level diagnosis is possible, single individual diagnosis is not. So what's the solution? Aaron outlined this eloquently. And his solution is that we unify structural and functional metrics, but now we add artificial intelligence to the solution to get this to work. And there's a terrific paper that shows that it can work. This is a paper in conventional MRI, normal appearing brain. So these are patients with schizophrenia, could be done in patients with autism where we don't see any abnormalities on conventional MRI. This came out of the Guangzhou Institute of Science and Technology in a collaboration with the Mayo Clinic. What did they do? Well, they did structural and functional MRI studies, and they developed metrics from both. They fed them into this machine. Now, this machine is a complicated machine. It has extreme learning, support vector machine, linear discriminant analysis, and random forest. And they fed these through and did some iteration. And what did they find in terms of diagnostic accuracy of concussion? It's astonishing. ROC values 99.3%. Problem is, in that black box, we have no idea what the AI was detecting. Now, I'm going to finish up here. I hope I have a little bit of time left, Carmela. Do I?
Sure, take a few minutes.
Talking about blood.
Sure.
Okay, thanks. So we have serum biomarkers. All of these have been studied. There is a summary of that in this test. It's called the C.E.T.S test. It was funded by the Department of Defense. What did they find? They found that is very good sensitivity. If you look at right across the board, patients with GCS 15, it had 100% sensitivity. Look at the specificity, 35.6%. That's not going to cut it for diagnostic tests. We're going to take it to the next step. And as much as we've looked at this and wondered about this, there is a recent publication that comes from Doug Fraser out at the University of Western Ontario. They were brilliant, I think, in this. They studied proteomics. They studied over 1,400 plasma proteins 72 hours after concussion. But it wasn't just studying the plasma proteins that was interesting. They used a new assay, which is tagging these antibodies to those plasma proteins with DNA, and they did a PCR amplification of this to detect very, very subtle injury. So out of those 1,472 plasma proteins, they found ATOX1, SPARC, and NT5C3A. ATOX1 is a cytosolic protein essential in the role of copper homeostasis. The brain has one of the highest levels of copper in the entire human body. The best performer is SPARC, which is a basement membrane protein expressed, guess where? In the endothelium and other cells. And it's also induced in mature blood vessels close to a site of injury. My goodness, this is about blood vessels, everybody. It's not about neurons. And the combination of the three led to an area under the curve that is virtually diagnostic of acute concussion in the first 72 hours. So in summary, and they also used the machine learning algorithm to sort out between those 1,400 proteins to figure out the best three. I think single subject diagnosis is clearly going to become feasible. And the other thing, it's going to allow us to assist linking the biology of concussion with physiology and recovery, especially versus persistence of symptoms. And I think you can add the neuropsychological features into this sophisticated AI machine as well. Ultimately, though, we have to make sure the tools can have a diagnostic success in individuals with comorbidities. So with that, it's springtime, and the future is bright, even though it was winter just 2 days ago when the temperature here was -2 centigrade. So thanks for your attention. I can take questions if there's time.
Thank you very much. Unfortunately, we don't have time for more questions, but
there's going to be a quiz after this because this was the review. So we make sure that all the attendants take the quiz.
Okay, great.